Hello, this video is intended for clients of the UCLA Lipidomics Lab. In this video, we will introduce some basic concepts in lipid biology and nomenclature, and then walk you through the various data and analyses you will be receiving from the UCLA Lipidomics Lab. If you are familiar with lipid biology and lipidomics, you can feel free to skip the first half of this video. In this video, and in the data you will receive from the UCLA Lipidomics Lab, we will be using the standardized lipid nomenclature and shorthand system adopted by the LipidMaps Consortium. We will provide an operational overview of this system. For further background, refer to this paper linked in the description. LipidMaps nomenclature groups lipids into 8 categories, listed here along with common examples. Our analysis will focus on the first 5, as they are abundant in mammalian samples. These categories are then subdivided into main classes and subclasses. The 17 subclasses currently measured by the UCLA Lipidomics Lab fit into the LipidMaps nomenclature as shown here. All of the species reported will use one of these shorthands, highlighted on the right. It should be noted that our coverage is not comprehensive. Currently, the LipidMaps database contains over 60,000 lipid species. We have targeted approximately 1,400 lipids that are known to be most abundant in mammalian cells, tissue, and plasma. In the average analysis, we report out several hundred of these species. The nomenclature for individual species within these subclasses will depend on the level of characterization within the mass spectrometry experiment. Here we will describe 3 levels of characterization as they relate to your lipidomics data. First is the basic species-level characterization, also known as bulk or chain composition characterization. This characterization gives you the total number of carbons and double bonds within species. However, for diacyl or triacyl lipids, this characterization is ambiguous as multiple acyl tail combinations could make up the species being measured. As an example, PC34-1 denotes a phosphatidylcholine with a total of 34 carbons and a single double bond on its 2 acyl tails. As shown here, there are a variety of acyl tail combinations that could be measured as such. If you search PC34-1 in the LipidMaps database, you'll be able to view other possible combinations. In our lipidomics assay, we report phosphatidic acids, PAs, with this bulk nomenclature. Most diacyl species reported in your lipidomics data provide acyl tail level characterization. This nomenclature denotes the number of carbons and double bonds on each acyl tail. As an example, PC160181 describes a phosphatidylcholine with a fatty acid containing 16 carbons and no double bonds, and one containing 18 carbons and 1 double bond. Within a phosphatidylcholine, these fatty acids are bound to one of the positions on the glycerol backbone, commonly called SN or stereospecific numbering positions. Within this nomenclature system, the underscore indicates that the position of the fatty acids is ambiguous. A search of PC160181 in the LipidMaps database will yield multiple results, including the 2 species listed here under fatty acyl position level. The slash indicates certainty as to the SN1 and SN2 positions of the 2 fatty acids. In the first case, the 16-0 fatty acid is in the SN1 position on the glycerol backbone, whereas in the second species, the 16-0 fatty acid is in the SN2 position. For some species like PC160181, our measurement is likely a combination of these 2 species. The determination of fatty acid position is another level of characterization. There are some cases within our assay where the positioning is certain, and those species will be annotated accordingly using a slash. These cases include ether or vinyl ether-linked phosphatidylethanolamines. It is worth noting that there are a few additional levels of characterization that are beyond the scope of our lipidomics assay. Our lipidomics assay does not differentiate between fatty acids with different double bond positions. As an example, here are 2 18-1 species that are common in mammalian samples. They differ in double bond position. oleic acid is an omega-9 fatty acid, whereas vicinic acid is an omega-7 fatty acid. These numbers indicate the position of the double bond relative to the end of the fatty acid chain. These two fatty acids also differ in their double bond configuration across the carbon backbone. Vicinic acid is a naturally occurring trans fatty acid, whereas oleic acid is a cis fatty acid. These two species come about by distinct biosynthetic processes. All of these bond position and configuration differences result in isomeric species that are measured together by the mass spec as 18-1. Another group of isomeric species that one should be aware of is that of straight and branch chain fatty acids. Here is one example of a straight and branch chain form of palmitic acid. Branch chain fatty acids are known to make up a minor component of many biological samples. As with the previous example, these structures have the exact same chemical formula and molecular weight. Thus, they are measured together by the mass spec. Now that we have reviewed the basics of lipid nomenclature, we will walk through a sample data set so that you'll know what to expect and how to begin analyzing your lipidomics data. For our example, we will be presenting lipidomic analysis of mouse macrophages. Much of this data can be found in this cell metabolism resource paper linked in the description. Upon completion of your lipidomics experiment, you will receive a zipped file containing your lipidomics data package. Each file will have the same root name, containing the date of the experiment, an arbitrary experiment number, and the name that you've given to your experiment. The primary data file is an Excel document containing just the root name. When we open this document, we see an Excel sheet with several tabs. We'll walk through these one at a time. On the left-hand side of each tab, you'll see the information from the sample map that you provided. This includes the sample ID, groupings of biological replicates, and the normalization values that you've specified. In this case, cell number in units of tens of millions of cells. Within this particular experiment, we are analyzing macrophages that have been stimulated with various TLR ligands. Within all sheets, you will notice that some cells are blank. It is important to note that these are not zeros, but an indication that these species are below the limit of detection or quantification. Within a shotgun lipidomics experiment, the detection of species is a function both of the abundance of that species and the other compounds in the sample. As such, a not detected value is likely lower in concentration than a reported value, but that determination cannot be made for certain. The first tab, Species Normalized, reports all species in nanomolar amounts normalized to the value you provided. In this case, that would be nanomoles per 10 million cells. If you scroll across this sheet, you'll see several hundred lipid species sorted in alphabetical order. Almost all of these species should be searchable on the LipidMaps database. Some may be missing as the database is a work in progress. As this large number of species may be difficult to work with, there are a number of ways that the data can be aggregated to help you understand the changes within your samples. The simplest is to sum all species within a subclass. This data is presented in the Class Normalized tab. As an example, we have detected 24 cholesterol ester species, and if we sum those together, we get the cholesterol ester value in the Class Normalized tab. For your convenience, we have plotted this data in the class total PDF files within your data package. Within this, and all other bar plots in the data package, the error bars are drawn based on one standard deviation. Here you can see the general profile changes within the lipidome of these macrophages. The TLR3 stimulated macrophages have large increases in cholesterol esters and triglycerides, while the TLR4 stimulated macrophages have the largest increases in phospholipids. Another aggregation sums together all species within a subclass that contain a specific fatty acid. As an example, if we look at the Fatty Acid Normalized tab, we can see amounts of phosphatidylcholine that contain one of 22 fatty acids in units of nanomoles per 10 million cell. As an example, you can see here that PCs containing 22,4 show the highest fold increase in the TLR3 stimulated condition. This value is a summation of 6 different 22,4 containing species that were detected in this experiment. The next 3 composition tabs in the Excel sheet provide an alternative analysis of your data. Rather than normalizing to the user-specified normalization values, these take the molar amounts of each lipid in the sample and present them in terms of a percentage. If you look at the Class Composition tab, you can see each subclass calculated as a percentage of all lipids measured. As an example, cholesterol ester accounts for 5 to 6% of all lipids measured in the TLR3 stimulated condition. On the Species Composition tab, each individual species is calculated as a percentage of that particular subclass. As an example, cholesterol ester 16-0 represents approximately 11% of all cholesterol esters in the TLR3 stimulated condition. This type of analysis can provide valuable insights. While all cholesterol esters are increased in absolute content in the TLR3 stimulated condition, the percentage of cholesterol ester 16-0 decreases in comparison to the not treated condition because other cholesterol esters show greater increases. Likewise, the Fatty Acid Composition tab calculates the percentage abundance of all species within a subclass that contain a specific fatty acid. As an example, 31% of all phosphatidylcholine measured contains a 16-0 in the TLR3 stimulated condition. The last 2 average tabs are drawn from the first tabs, Species Normalization and Class Normalization. In these tabs, we have calculated the average and standard deviation for the groups of biological replicates as assigned by the user in the sample submission form. These sheets are produced for your convenience and can facilitate the rapid generation of plots in Excel. Our standard data package also includes a heat map and a principal component analysis. The PCA plot can help you visualize the differences between the groups in your experiment. In this example, TLR4 stimulation shifts the lipidome on PC1, while TLR3 stimulation shifts the lipidome on PC2. The PCA loadings are an excellent resource that can direct you to the individual species that drive these shifts. The species labeling on the heat map is often too small to be read, but the general profile of the heat map can help you identify potential patterns and outliers within your data set and groups. In this example, you can see that all groups cluster together and there don't seem to be any obvious outliers. Within our analysis, triacylglycerides are measured differently from other subclasses. Because they have 3 acyl tails, we do not identify all 3 acyl tails. We measure the bulk number of carbons and double bonds on all 3 fatty acids. We are then able to fragment off and identify 1 fatty acyl tail. For each bulk species, we carry out this fragmentation for all likely fatty acyl tails. In this example, you can see that we measure 9 likely acyl tails on this bulk species. This level of analysis results in the reporting of hundreds of TGs in the average sample. One way to aggregate and simplify this data is to sum all bulk species containing a given carbon or double bond number. This data can be used to create 2 plots that give a general profile of the TG pool. If you look at the double bond plot from our example experiment, you can see a striking pattern. While both TLR3 and TLR4 stimulation results in an increase in total TGs, the profile looks very different. TLR4 stimulation most profoundly increases TGs containing 1, 2, or 3 double bonds total, corresponding with monounsaturated fatty acids. TLR3 stimulation results in the most significant increases in TGs containing greater than 4 double bonds, corresponding with polyunsaturated fatty acids. This corresponds with an overall shift in the total carbon length. As TGs are a store of lipids, monitoring these gross shifts in pool makeup can often yield insights. These plots and the summation data used to create them are in your data package. One other caveat worth mentioning. While we report free fatty acids within a sample, it should be noted that these measurements are never perfect. Free fatty acids are found ubiquitously, even in the highest grade of chemical solvents, within plastics, and nearly everywhere else in nature. As such, the mass spec assay will measure some baseline level of these, especially saturated fatty acids, in combination with the actual fatty acids from the sample. This fact may distort the magnitude of changes. If free fatty acid measurements are critical to your experiment, let us know so that we can discuss this further. This concludes our walkthrough of your data package. If you have further questions, please email us to set up a consultation.